a very, very special guest. This is Yannick Duwe, who was the first child enrolled in our study. He was 9 years old when he enrolled. He's now 15. And he has a twin sister who's unaffected, who is here as well, and his mom is here as well. And his father, Tony, is sitting right next to him. And Yannick is just an extraordinary person. He wanted to learn all about the scientific rationale for the study, and in fact, after his injection, he told us at age 9 years old that he wanted to become an ophthalmologist when he grew up. I don't know if he still does. But Yannick, what do you remember about the process in deciding to enroll in the study? Was it scary? Would you do it again?
Well, I don't remember much, but yeah, yes, I want to do it again.
What do you remember what it was like before you had the intervention compared to your vision now?
I remember in parts, but it was very difficult in some things. But I was very young, and it's not so clear.
And what advice would you give other children who are deciding whether or not they would enter a gene therapy clinical trial?
The advice that I give is just do it.
What was it like at school when you told people you had to travel to the United States?
At school, I worked before in Braille, and after the surgery, I began to work at the computer, and it's very much easier than in Braille. I can work at higher speed now, and it's very easy now.
Fantastic. And I would like to now open up the session for questions from anybody in the audience, as well as the panel members, for any of us, and for Yannick Duwe and his father. Actually, let me ask, while you're deciding about your questions, and perhaps the room lights could go on, I'd like to ask his father, Tony Duwe, what do you remember about the whole experience?
I remember everything. I was not 9 years old, but for us it was relief that we can come to Philadelphia, to the shop, because we were searching 6 years for a cure to help my son, and we know that when there was nothing that we can do, he became completely blind. So when he was 9 years, we met you, and now he's seeing much more, and he is more independent than before. Before we must help him with everything he would do, and after the first surgery, after 4 weeks, he can do more things independent, and for us it was a complete other world. So thank you again for everything.
Thank you, the team effort. Yes, there's a question here.
This is not a question, but I am from the patient community and the father of a 20-year-old son with inherited retinal disorder. And I just want to say that, us in the patient community, you see some people do incredible things like run 12 marathons in 1 year or do an Ironman, but we do this because we have to. And I just want to say that the people on the stage and everyone in this room has options on what you're going to do. So just from the patient community, I want to let everyone know just how deeply appreciative we are of the late nights, the hard work, the frustrations that come with this. But again, you do this as a choice, and we just appreciate it beyond words can express.
Thank you.
Jane, for those of us that work with dystrophies, this has been fabulous work to follow, and your explanations here have shown us much about the complexity at the technological level and at the regulatory level. What I think many of us now are asking, as you well know, is that more than 50 genes cause a wide range of dystrophies of the retina and the macula. There's no way that we can spend the 5 to 10 years of basic science and 15 plus years of clinical trials for each of these if we're going to manage this kind of blindness. What do you see now with your experience, both at the technological level and the regulatory level, of being able to treat these orphan retinal diseases?
Well, I'll start an answer, and perhaps my colleagues would like to pipe in. That, of course, is a really incredibly important question. And so our view of this, or at least my view of this, is that the RP65 form of LCA is a stepping stone. That through this study, we are obtaining safety data and the know-how of how to move forward with clinical trials and define relevant outcome measures so that that information can be leveraged to be able to develop faster treatments for other retinal degenerations or other disease genes. And for some of these, I think it's going to be fairly easy. So for example, our team is leveraging the data from the RP65 studies to be able to move to choroideremia next because the vector is very similar, et cetera. The delivery is similar. But for others, it will require a little bit more basic science. But I think now with iPS cell work and so forth, it could be sped up.
A major part of it is the regulatory one. Will we have to go through this entire process for each gene, or be able to get some blanket means of using the technology for a variety of genes?
Well, so I don't know if any of our regulatory colleagues are in the audience and would like to address that question, but I think that from our perspective, we would certainly believe that the course and the time course of a subsequent trial could be considerably shortened. I mean, for example, there was a concern in the first trial about whether it would be safe to inject the contralateral eye, and I think that that's, now we have data for this vector about that, and so I think, one can see many places in the timeline that can be shortened going forward for subsequent applications. But obviously you've touched on a very important issue, and both investigators and the regulators are interested in shorter timelines to registration.
Question?
Yes, thank you. Thank you. Thank you, Dr. Bennett and the panel, for your wonderful presentation. How soon after phase 3 is complete do you think gene therapy will be FDA approved and ready for general use?
Well, so I think that experience in many different areas of drug development would say that from the conclusion of phase 3, from the readout of the data, to preparation of a biological licensing application would typically occupy 6 to 9 months, and then once the BLA is submitted to the FDA or the marketing authorization application to the European Medicines Agency, they have 6 to 9 months to examine the application and make a decision. And typically that takes place in the context of an FDA advisory committee meeting where experts in the area come together and review the data and make a recommendation, as well as, of course, the final decision authority is with the regulators. So, I think we have to be honest to say that even after the conclusion of the study, a number of months will pass before there'll be a decision.
So